Welcome together. My part focuses on the functional characterization of proton-coupled sugar transporters. The data I will present is from my PhD thesis I did in Klaus Fendler's lab at the Max Planck Institute for Biophysics in Frankfurt. And I have to point out that I used the custom-built instrument during my work. During my PhD, I investigated 4 different proton-coupled sugar transporters from bacteria with known structure, mainly using SSM-based electrophysiology. I purified the proteins and reconstituted them into proteoliposomes. This way I got a sample with large protein density, generating huge current amplitudes on the SSM. There already have been ideas about the basic principle of transport. The transporters exist in different conformations, as you can already see in the solved structures. There are outward-facing and inward-facing structures, observed for fugue P and lug Y respectively, as well as occluded structures. The transport most likely occurs by large conformational transitions to alternatively expose the substrate binding sites to the inside and the outside of the cell. This principle is called alternating access. For proton-coupled sugar transporters, the following mechanism was proposed. In the outside-facing conformation, proton binds to the protein, which may increase the affinity for the sugar. Sugar binding then triggers the conformational transition to the outside-facing conformation. This is followed by sugar and proton release, and the following conformational transition of the empty carrier back to the outward-facing conformation. It turned out that different reactions of this reaction cycle can be detected by SSM-based electrophysiology. The different reactions in this transport cycle depend on the pH and sugar concentrations inside and outside of the cell, as well as the membrane potential. These parameters therefore can be changed in the experiment to conclude details about the transport mechanism. I started to investigate the pH dependence of the different transporters. For the 3 E. coli sugar transporters shown here, the transport is decreased in the acidic and alkaline pH range. The acidic pH range is shown on the top, the alkaline pH range is shown at the bottom. In the neutral pH range, all transporters show a high transport activity. When plotting the peak currents against the pH, bell-shaped curves are obtained, showing an acidic pKa and an alkaline pKb. Here on the left side, the acidic pH range with the 3 pKa's of the transporters are shown, and on the right side, the alkaline pH range and the pKb values are shown. In our kinetic model, this would make sense, assuming that proton depletion inhibits proton binding, and therefore overall transport activity in the alkaline pH range is decreased. On the other hand, proton excess in the acidic pH range inhibits intraliposomal proton release, which under these conditions becomes a rate-limiting step. Transport is also decreased. If this is correct, the alkaline downregulation must be due to the extraliposomal pH, where proton binding occurs, and the acidic downregulation must be due to the intraliposomal pH, where proton release occurs. So we had to measure under asymmetrical pH conditions, meaning different pH values inside and outside the proteoliposomes. This can be easily done by using solutions of different pH values, here depicted as blue and yellow. Incubation of several minutes at the yellow pH yields intraliposomal pH equilibration. Before activating the transporter using the activating solution, the liposomes are facing the blue pH only for 1 to 2 seconds. Therefore, the measurement takes place under pH gradient conditions. Let's start with experiments using pH gradients close to the acidic pKa. The question is, does the pKa reflect intraliposomal proton release? Or in other words, does transport activity depend on the intraliposomal pH? Or is intraliposomal proton excess responsible for the downregulation of transport? Before starting with electrophysiological experiments, I had a look at proton diffusion across membranes. Proton gradients are not stable over a long period of time, since all membranes are more or less leaky for protons. I did measurements using the pH-sensitive fluorophorpyramine. I trapped pyramine into liposomes and proteoliposomes of different lipid compositions, pH titrated them, and looked at the change of intraliposomal pH. Thanks to these experiments, I knew the kinetics of intraliposomal pH change. It's biphasic with distinct time constants depending on the liposome composition. Then I used the same proteoliposomes and did the SSM experiment. I made a solution exchange from acidic pH to neutral pH, followed by activation of the transporter and measuring the transport activity. The seconds after pH change, the activity of the transporter was still low, followed by the increase of the P currents. Interestingly, the time dependence of the change in transport activity after pH titration fits with the kinetics of proton leakage through the membrane, so transport activity perfectly follows the intraliposomal pH change. This proves very well that the change in transport activity is due to the intraliposomal pH change, and the proton release reaction becomes rate limiting below the pKa. Now let's see how the transport activity responses to a pH change close to the pKb. Here our assumption was proton depletion slows down proton binding to the outward-facing transporter conformation. This means only the outer pH should affect transport activity, which should be changed only milliseconds after the buffers of different pH have been exchanged. I did the same experiment as shown for the acidic pH range using the alkaline pH range. I don't show you a time dependence here because there is no time dependence. Instead, I show you the transient currents before and 1 second after the respective pH jump. As you can see, under symmetrical pH conditions, you observe a large P current in the neutral pH range and a small P current at the alkaline pH range. 1 second after switching from alkaline to neutral pH, the transport activity has fully recovered. As a short reminder, in the acidic pH range, this took over 20 minutes due to the intraliposomal pH change. The same is true for the opposite experiment. 1 second after switching from neutral to alkaline pH, the signal has fully decreased. Therefore, indeed, only the extraliposomal pH is relevant for the alkaline downregulation, and proton binding becomes rate limiting under these conditions. Besides the pH dependence of steady state, I observed another interesting aspect related to pH. When measuring under extreme pH conditions where steady state is inhibited, we still could observe sugar-induced currents. They are much faster and do not reflect transport, but a sugar-induced electrogenic pre-steady state reaction. I will refer to these as sugar binding currents. These currents are also much smaller, so the currents shown here are normalized to the respective P current. The observation of sugar-induced currents means that the transporter still binds sugar under extreme pH conditions. I should also point out SSM-based electrophysiology is sensitive enough to measure fast events with low electrogenicity, such as sugar binding. There are also mutants which are unable to bind protons due to the lack of the proton binding site. In these mutants, only sugar binding currents and no steady-state currents are observed. Since in wild-type sugar binding currents are not visible between pH 4 and pH 10 due to much more prominent steady-state currents, the mutants allow for the investigation of the pH dependence of these sugar binding currents. In contrast to the bell-shaped pH dependence of the steady-state currents, reflecting proton binding and proton release, the pH dependence of sugar binding is linear. It cannot be described by the titration of one or more proton binding sites. It must be an environmental effect. Maybe the membrane surface charge gradually affects a sugar-induced electrogenic conformational transition. Sugar binding currents so far only as a side note and an example for the possibilities of SSM-based electrophysiology. But now I will switch to GLCP. In literature, this transporter is described as a proton-coupled glucose transporter, despite being homolog to the human glucose transporters, which are uniporters and not coupled to proton translocation. We had a look at the pH dependence and tried to reproduce the bell-shaped dependence we have been familiar with. Instead, we only observed sugar-binding currents for all pH values. The graphs show a comparison of the currents in GLCP and xylE. xylE shows typical slow transient transport currents, while GLCP only shows fast sugar-binding currents. In addition, we recorded traces for both GLCP and xylE using proteoliposomes with different lipid-to-protein ratios. Only for steady-state currents, a higher protein density yields faster decaying currents, as observed for xylE but not GLCP. GLCP, therefore, is a special case, showing no proton transport under these conditions. Here's the full pH dependence shown. It doesn't look like the bell-shaped pH dependence of wild-type E. coli transporters, also not like the linear pH dependence of the sugar-binding currents of transport-deficient mutants. We observed a single titration curve with 1 pK. Also, the polarity switches from positive to negative up on deprotonation. From literature, we knew the proton binding site in GLCP should be D22. So we used a mutant with a mutated proton binding site and found that the currents for all investigated pH values are positive and no pK can be observed anymore. This shows that the deprotonation of D22 is responsible for the switch in polarity of the signals in wild-type GLCP at pH 8.6. What does it mean? We thought that above pH 8.6, the sugar induces a conformational transition which exposes the negatively charged proton binding site to the other side of the membrane. Maybe also the sugar is released, which would mean there is uniport of the sugar molecule. With electrophysiology, we only could observe proton movement. To look at the sugar movement, we performed a radioactive transport assay. We compared the sugar exchange activity at pH values below the pK and above the pK, meaning we investigated sugar translocation in the protonated and unprotonated transporter. Interestingly, there is no pH dependence. The sugar is transported into the liposomes independent of pH. This is a kind of crazy property for a transporter described as proton-coupled. Substrate translocation independent of the protonation state. GLCP therefore must likely perform uniport independent of pH. Then we asked ourselves, maybe for symport to cure, we need the protons as a second driving force, so we did measurements on the SSM with pH gradients. We applied an inward-directed pH gradient, followed by the activation of transport by a sugar concentration jump. Compared to the signal without a pH gradient, shown in black, the signal shape differed a lot. With a pH gradient, the trace here is shown in red, the signal got broader, an indication for stationary transport activity. Indeed, after reconstruction of the transporter currents, we could clearly observe stationary sugar-induced proton translocation, a phenomenon known as symport activity. Interesting enough, I just wanted to finish with a negative control. I applied the opposite pH gradient. I thought application of an outward-directed pH gradient before activation with an inward-directed sugar gradient would again diminish the stationary currents. But what I saw were stationary currents in the opposite direction, meaning sugar-induced proton efflux, also known as antiport. Of course, the observed transport modes shouldn't be called symport and antiport, since both modes mechanistically cannot occur in the same transporter. Instead, GLCP is a loosely coupled transporter. Sugar can be transported alone, but if you have a proton gradient, sugar translocation is increased. We used an 8-state kinetic model where all described transport modes are possible, and we could simulate our experimental observations in silico. We could also show that due to the membrane potential in vivo, the loosely coupled system is sufficient to facilitate sugar import. Strict coupling is just not necessary. At the end of my presentation, I just wanted to highlight a few more datasets I could generate using SSM-based electrophysiology. Of course, we had a look at substrate specificity and measured currents by application of different substrates and substrate concentrations. Using the wild-type transporters, we determined Km values for different sugars. It's important to know that SSM-based electrophysiology cannot determine absolute turnover rate in molecules per second, since the amount of transporters on the sensor is unknown. But we could compare relative Vmax between different substrates. Regarding substrate specificity, like Y and P behave differently. The respective main substrates are highlighted red in the table. Like Y shows the highest Vmax but lowest affinity for the main substrate lactose. In contrast, P shows the highest affinity but lowest Vmax for the main substrate fructose. Transporters can either optimize substrate translocation by increasing the turnover or affinity. Also, we showed that some substrates show a hill-like substrate concentration dependence, indicating different transport mechanisms between substrates. Using the transport-efficient mutants, we could determine Kd values for sugar binding. From pre-steady-state currents, we can also derive rate currents of the reaction. In this case, the sugar-induced reaction has a rate constant most likely above 300 per second, since the currents are likely limited by the time resolution of the instrument. Last but not least, we can also investigate the crosstalk between sugar binding and protonation and gather information about the cooperativity. For the 3 proton-coupled sugar transporters from E. coli, with decreasing pH, the sugar affinity is increased. This makes sense. Protonation facilitates sugar binding. Positive cooperativity is a property of this importer. Interestingly, in GLCP, the loosely coupled transporter, we observed the opposite. The protonated transporter has a lower sugar affinity than the deprotonated transporter. This is a property of an antiport and may be an indication that antiport is favored in GLCP. Let's summarize our findings. We could show that the proton-coupled sugar transporters have 2 different pKs assigned to the proton binding and the proton release. Since there is only one proton binding site, for each transporter we suggest a pK switch as a central mechanism for the import activity. We found that GLCP represents a loosely coupled transporter. It could be viewed as an evolutionary state between the strictly coupled symporters like SILE and the human glucose transporters of the GLUT family, which lack the proton binding site and facilitate only sugar uniport. Another interesting common phenomenon among the sugar transporters is that sugar binding can be observed directly. Finally, we characterized the transporters regarding the substrate specificities and cooperativity. That was my part of the webinar. If you have any questions, just type them into the Q&A chat and we will try to answer at the end of the webinar. I also want to thank my PhD supervisor, Klaus Fendler, for the continued support during and even after my PhD.